Hi, I'm Troy. If you want to know how to remove cancer-causing chemicals from your blood, stay tuned. I've been doing a detox experiment on myself for almost 5 years now, and I've broken the world record for removing blood lipid PCBs and DDT. Mainstream medicine says there is no known means of removing these chemicals from our blood, but I've found a way to do it. I'm here today to share with you the basics of my breakthrough discovery. To begin with, PCBs are indestructible, carcinogenic chemicals that were produced in the 20th century to do many things, mostly act as insulators inside transformers. DDT is another practically indestructible chemical. DDT was used in the 20th century as a pesticide. DDT breaks down slightly into a chemical called 4,4-DDE. These chemicals are in the foods we eat, and they bioaccumulate in our fat cells. When they reach a certain level, they can trigger a cancer response. Babies often have very high levels of these chemicals because they absorb them from their mother's blood and breast milk. High levels of these and other chemicals definitely increase a child's risk of developing cancer. This makes it imperative that we find a way to detoxify ourselves so we don't pass these concentrated toxicants on to our offspring. I happen to have a lot of experience with extended fasting, so I took it upon myself to try and prove that this ancient practice of not eating for long periods of time could actually do what they said couldn't be done: detoxify the body of these carcinogenic forever chemicals. I started my experiment in May of 2015 by giving blood for a baseline sample. I sent my blood serum to a cutting-edge lab for extremely expensive mass spectrometer tests. In the summer of 2015, I fasted for 21 days. By the final day, I had lost a total of 31 pounds. I did a number of other interventions, including saunas, taking supplements, etc., all of which I recorded in my book. My initial DDE level was 56,100 parts per trillion. By January of 2016, I did my second blood drawing and found that DDE levels had dropped 12% to 49,100 parts per trillion. My initial PCB level was 71,100 parts per trillion. PCB levels actually increased slightly by January to 71,900. This increase, I presume, was because of the way the fat cells excrete toxicants when you fast, like a sponge being squeezed out. I decided to continue taking blood samples every 6 months or so in the hopes that the levels would continue to fall. In 2016, I discovered a weakness in my method that forced me to change my test protocols. To explain, the liver produces an enzyme called cytochrome P450, or CYP3A4 for short, and this is actually able to identify chemicals that don't belong in the body. CYP3A4 identifies the chemicals that don't belong in the body and tags them with an oxygen atom for elimination. These tagged chemicals are shunted off into the bile and deposited in the small intestine for elimination. Unfortunately, the small intestine can't filter them out and inadvertently reabsorbs these cancer-causing chemicals back into the bloodstream. This practically endless loop is called the enterohepatic pathway. To attempt to interrupt this cycle, I began taking activated charcoal in August of 2016. The idea is that before these chemicals can be reabsorbed by the small intestine, some of them will be adsorbed onto the highly corrugated surface of the activated charcoal and carried outside the body. The DDE blood test results from July of 2016 were almost unchanged at 48,600 parts per trillion. My blood test results from the January 2017 blood drawing showed 44,500 parts per trillion, an additional 7% drop. My cumulative decrease for DDE was 20.68%. For PCBs, the July 2016 level was 64,400 parts per trillion, a drop of 9% from baseline. My January 2017 PCB results were 59,700 parts per trillion, an additional 6% drop. which seemed to suggest that the activated charcoal, begun here, was having an effect. My cumulative decrease for PCBs was a 15.92% drop from baseline. To try and get better results, I decided to keep taking activated charcoal and do 2 more fasts of longer duration. In the summer of 2017, I did a 30-day fast. Here I am on day 4. Here I am on day 16. Getting thinner on day 22. Day 26, down to 154.5 pounds. Here I am on day 30 at 151 pounds. By the end, I lost a total of 34 pounds, but was still able to walk around and drive and participate in events. In the summer of 2018, I did a 40-day fast. This is me on day 7. On day 14, I weighed 166 pounds. On day 22, I was 160 pounds. By day 28, I was down to 156.5 pounds. Here's a selfie from day 35, still able to drive around, but looking pretty gaunt. Here's day 40, hallelujah, I made it to the end. I had lost a total of 44 pounds. During this time, I did not do the saunas or take supplements like I did back in 2015 in order to simplify the experiment with fewer interventions. I had been taking blood samples every 6 months, and I sent them all to be tested in December of 2018. I received the results in April of 2019, and they were surprising to say the least. First of all, the blood sample from June 2017, prior to my summer fast, showed a huge, unexpected drop in both chemicals. The DDE levels were 22,600 parts per trillion. This is a 39% additional drop and a 59.71 total drop from baseline. PCB levels also dropped a huge amount at this time. They were 30,500 parts per trillion in the June 2017 measurement. This is over a 40% additional drop and a 57% drop from baseline. My best hypothesis to explain these huge drops is that my 21-day fast from back in 2015 must have triggered altered gene expression, which lasted for over a year and was finally wearing off as I reached the 2-year anniversary of the fast. The cells were then reverting to their default settings and a new equilibrium was reached, showing how much detoxification had actually taken place. Going back to the DDE chart, the second big surprise was that there were significant elevations in the chemicals after these fasts. It could be that these are due to the greater lengths of the fast, or perhaps because in 2017 and 2018 I did not do the additional detox protocols like saunas and supplements. In the 2015 fast and the period following, I did over 100 saunas, so this could have actually made a very big difference. Levels for DDE dropped to their lowest point as measured in June 2018 of 22,000 parts per trillion, which is a cumulative drop of 60.78% from baseline. This is definitely a world record. Going to the PCB chart again, you can see again we have the spikes after the fasts as on the DDE chart, and the lowest point also was in June of 2018, where we have a measurement of 29,200 parts per trillion for PCBs, which is a 58.87% cumulative drop. Again, definitely a world record. Notice that for both chemicals, although the fast done in 2018 was 1/3 longer than the fast done in 2017, the amount of the spike that follows the fast is 1/3 as high in 2018 as in 2017, which strongly suggests that toxicant levels have decreased a great deal. And if we look at the 2-year time following the original fast before the giant drop, we would expect to see something similar happen 2 years after the 2018 fast, which would take us to summer of 2020. All this seems to indicate that a great deal of cleansing had taken place, and all evidence suggests that a much bigger drop is around the corner, considering the giant previous drop after the delay of 2 years. One huge question remains as to the effectiveness of taking activated charcoal. I currently have a friend doing an experiment on himself with a single intervention of taking activated charcoal, so we will hopefully have some evidence soon to help resolve this question. My last blood drawing was in December of 2019, and now I'm awaiting results from the lab. In the meantime, for those of you who don't want to get cancer and don't want to pass on cancer-causing chemicals to your kids, I've put everything I know about detox into a book, which, for a limited time only, I'm giving away free twice a month in electronic form as a promotion to get people to read it, talk about it, and review it. My book is called Detox Fast. You can find it on Amazon, and I'm making it free for ebook download on the 1st and 15th of every month till further notice. Detox Fast includes the history of fasting, the physiology of fasting, experiments on fasting, all my experiences in detail, safety precautions, day-to-day journals, complete blood test results, a brief history of toxicity, ways to avoid toxins and toxicants. Again, the book is called Detox Fast. For a limited time only, you can download the ebook free from Amazon on the 1st and 15th of the month. If you like the book, please leave a review and spread the word. To find out more, see my website at troyricharder.net. Also check out my nonprofit's webpage at hhresearch.org. Thanks for watching.